How can astronomy help us understand the spread of COVID-19 and other diseases? Hey team, I'm Chris, and I want to tell you about a cool project I've been working on for the last few months. I have a PhD in cosmology, so you'd usually find me doing research about the physics of the universe in the first fraction of a second after the Big Bang at the Institute of Cosmology and Gravitation, which is part of the University of Portsmouth. However, I've recently been working on a very different project with a group of researchers still at the University of Portsmouth to try to better understand the spread of diseases like COVID-19. The work that led to the technique that I want to talk about in this video has actually just been published by our group in this paper here. So what does cosmology have to do with the spread of diseases? How can we use a piece of computer code designed to find stars and galaxies in the night sky to instead track the spread of viruses through coughs and sneezes? So let's start with how diseases like COVID-19 spread. The current World Health Organization advice says that COVID-19 primarily spreads through respiratory droplets and contact. This means that when you breathe out or cough or sneeze, bits of spit and all sorts fly out of your mouth, and these can potentially carry with them the virus. Now the drops that come out of your mouth when you cough or sneeze, they act like little swimming pools for the virus to live in and spread. The technical name for these droplets is viral-laden mucosalivatory respiratory droplets. But that's a bit of a mouthful, so I'm just going to call them droplets here. If these droplets land on a person, say on their face, the virus can find its way into their body and infect this new person, who I think should be called the splatee. A more likely situation when you cough is that your droplets land on a hard surface, and then someone else comes along, touches the surface, gets the virus on their hand, touches their own face, and viruses can spread that way. So it's super important to understand how these droplets travel through the air when we cough or sneeze. How far can they go? Where do they land? And how can we use this information to help stop the spread of diseases? When we cough, tiny particles called aerosols are also produced. These aerosols are very light and they tend to stay airborne for a very long time, whereas droplets tend to follow a sort of projectile motion and fall to the ground under gravity in quite a predictable way. It's possible that these aerosols can also carry the virus, but this seems to be a less common source of transmission at the moment. However, the technique I want to talk about here isn't sensitive at all to aerosols, so for the rest of this video, I'm only going to be talking about the spread of disease through larger droplets. So, how does astrophysics come into this? To start with, let me show you a piece of software that's used very commonly in astronomy. It's called Source Extractor, and it's commonly used to find things like galaxies and stars and other interesting objects in photos of the night sky that we might find interesting and want to study. For example, let's take a look at this photo taken by the Hubble Space Telescope. Now this is actually a very small section of the famous Hubble Deep Field photograph. In this picture, each bright spot isn't a star, but rather an entire galaxy, each containing billions of stars. If you ever want to feel small and insignificant, this is one of the best photos to have a look at. Now this piece of software, Source Extractor, allows us to find bright objects in pictures like this. So let's have a go at running it on this image and see what happens. Since this is a crowded image already, let's ask the code to find the 100 biggest and brightest objects in the picture and draw a nice ellipse around them so we can easily find them. If we run that code, we get a picture that looks like this. You can see the code's done a really good job at finding objects and drawing nice ellipses around them, even when, for example, here, when they're overlapping, or here when there's a small object right next to a bigger object. It still does a really good job of finding these objects, picking them out as two different things, and drawing a nice ellipse around them for us. One thing to note is the code actually draws these ellipses a little bit bigger than the objects it found, and that's just to help guide our eye so that we can see things easily in the picture. Often, if we have a picture that's a lot darker than this one, the code will actually be able to find things that are too faint for human eyes to see. This is a pretty useful tool for astronomy and astrophysics, but how on earth is it useful to study the spread of diseases like COVID-19? So we know COVID-19 spreads mainly through droplet transmission, and when we cough or sneeze, these droplets are most likely going to be your spit. So to understand the spread of diseases, it would be helpful to know where all the spit goes when you cough or when you sneeze. But in general, it's pretty difficult to see spit clearly. I mean, if you were to just cough in a room, it'd be very difficult to follow every drop of spit and to see where it lands in the room. As we're going to see, when you cough, a lot of spit comes out, so there's a lot of droplets flying around the place. And if you want to find and count each of these by hand, it's going to be very difficult and take a very long time. So how can we make it a bit easier to spot the spit that comes out of your mouth? One option is to put a bunch of fluorescent dye in someone's mouth. So when they cough, we get a beautiful spray of brightly coloured spit, rather than a boring cloud of hard to see dribble. When we did this, we used a fluorescent dye called fluorescein, which glows really nicely under a UV light. You can see here that I've got a bag full of the dye, and it's glowing really nicely under a UV light, so this should be really easy to spot if someone coughs this everywhere. This dye is commonly used by ophthalmologists, and usually they'll put it in your eye to try and find either an object in your eye or damage to your eyeball itself. But fluorescein dye is also safe to put in your mouth, so we can safely use it to colour the droplets of spit in a cough. So let's take a syringe of this dye and put it in our willing cougher's mouth. We use around 2.5 milliliters of saline, which is just salty water to mimic the spit, and we dilute about 1/20th of that amount of fluorescein in with the water. And this amount is about what you'd expect for someone with a respiratory illness to cough out. Like I say, we used saline the first time we did this, but you can actually get synthetic saliva made. So in future iterations, that's what we're going to use. Once our cougher is fully loaded with fluorescein, we simply get them to cough straight forward. You can see this beautiful spray of bright yellow spit, which is now super easy to see, traveling everywhere. This picture is quite a powerful reminder that every time you cough or you sneeze, a lot of spit comes out of your mouth, and it goes everywhere. And it's one of the reasons that simple face coverings are so effective at helping reduce the spread of viruses. Every one of those drops of spit could potentially carry the virus in it. So if it's caught in a face covering, it can't land on someone else or on another surface and spread the infection around. We want to repeat this experiment a lot of times, so we actually don't use a human cougher for this. We use this machine here, which is sort of a cough simulator. You can see here that after a couple of coughs, even the nozzle of the machine is glowing really brightly due to the fluorescein. We use this machine, the cough simulator, for a couple of reasons. Firstly, most places where we want to do this experiment aren't super keen on having a bunch of us come in and cough a whole load of times in their buildings during a pandemic. Secondly, we want a nice consistent cough speed and direction, and this machine tends to be a lot better than most people are at being consistent with the speed and direction. In the future, we of course do want to use humans as well, because that's what's really spreading the disease, and in the future we will do this experiment again with humans. Okay, so now we have a nice way of making the spit visible from a cough. So what we do is we set out a track of targets so that we can catch the spit and easily see where it lands. To do this, we just set out a track of 10 A4 sheets of paper, and on this piece of paper I'll print just a border and a ruler, and they're set out in front of the cough machine in a straight line. The rulers and templates are helpful so we can make sure we can accurately find the targets in photos of this setup, and the rulers help us confirm how big the droplets are in real space. So you can see the full setup of our experiment here. We have the cough simulator on this side, and in front of it 10 sheets of A4 paper which act as targets to catch the spit. And 10 sheets of A4 covers about 3 meters. One thing to note here is that our target track is quite thin. It's just the width of an A4 piece of paper. So when the spray goes to the side as well, we do miss that in this version of the experiment. This can be mitigated in the future by just having a wider track, or even covering the whole room in sheets and imaging the entire area of the room. We then photograph each of these target sheets and we get pictures that look a bit like this. This photo was taken under UV light, which is why the paper looks blue rather than white, and why the fluorescein is glowing its nice yellow colour. So in this picture, any spot that's yellow rather than blue is where spit would have landed, and where the virus could potentially be living on a surface, waiting to be spread. So now we have some nice pictures where we can easily see where the spit has landed. And we want to use this together with Source Extractor, that piece of code that finds galaxies in the night sky, to understand more about the travel of diseases. This is the stage that I think is really cool. If we run this image through a filter that basically turns the yellow into white and the blue into black, then something really amazing happens. The image of the fluorescent spit now looks exactly like a picture of the night sky. This is the sort of picture you'd expect a telescope to produce, but in actual fact, it's galaxies of drool swimming through a background of UV lit paper. So I think this is really amazing, and it probably gives you a hint as to what we're going to do next. But the first thing we do is actually crop out all the bits we don't care about. The only thing we're interested in this picture is the actual sheet of A4 paper. We don't care about what's on the outside of it. So now we take this image and we just run it through a piece of code that finds the A4 template and crops out everything else that we don't care about. So this is what the code produces for the picture I just showed you. I'm just showing you the blue and yellow image here because it's a little bit easier to see the edge of the paper in this case, and you can see that the code does a really good job of finding that A4 piece of paper and cropping out everything else. During this stage, we also de-warp the image so that we have a nice face-on view of the piece of paper. If you take an image that isn't straight on, you get warping in your picture based on the angle you're at and the distance you are away from your subject, and this makes things look a different size and shape than they are in real life. So the code actually takes that into account when it de-warps this picture. The template on each of the target sheets actually makes this automatic finding the paper and the de-warping easier, which is why we print it on every sheet along with a ruler. The A4 sheet has been found and cropped, and the image has been de-warped, so we're now looking at it straight on. This is what the image looks like in blue and yellow, and if you want to see it in black and white star mode, this is what you get. At this stage, it's quite hard to tell the difference between someone's disgusting cough and the beautiful night sky above you, which is something that I think is really cool at this point. Remember that for each cough we perform, we actually have 10 of these sheets covered in spit. And actually, we've managed to automate this whole process, so it's actually quite quick to analyze all the pictures we take, and if we were doing it by hand, this would be an almost impossible task. Okay, this is the point we've been waiting for. We can now run the Source Extractor code on this image, and it works exactly the same way as it would if it was looking for galaxies in the sky. It finds the bright spots on the dark background, except this time it's finding spots of spit on a piece of paper instead of objects in the night sky. So if we run the code on this picture, this is what we get. And again, I'm showing you the blue and yellow version just because it's a little bit easier to see. Every orange ellipse in this picture is where a spot of spit on that piece of paper has been found by the code. So we can easily see that the code does a great job of finding those big spots that we can see by eye. The other thing that's really clear in this image is how many teeny tiny drops the code also detects. Remember that each one of those represents a tiny spot of spit that the code has found on this piece of paper. To me, it's really surprising how many tiny drops of spit really do get produced every time you cough, and seeing it laid out like this is quite shocking. So for this piece of paper, let's think about how big the spots are and how many of each size that we can see. We can see there's a few quite big spots on this sheet of paper, and we also see that there's loads and loads of the tiny spots of spit. This is because this sheet of paper was pretty close to the cough simulator. It was about half a meter away from it. Let's compare this to a sheet of paper that was a lot further away from the cough. So this sheet was about 2.5 meters away from the cough, and we can see some pretty striking differences. First of all, there's a clear reduction in the number of spots we see. At this distance, we see that none of the teeny tiny drops make it this far, but there are still a few of the bigger droplets that do make it this distance. Since the droplets here have a much larger volume than the small drops that don't travel as far, they can still carry a lot of virus with them, and so there's still quite a big risk at transmitting the disease, even at such a large distance. Remember, this sheet was 2.5 meters away from the cough. This is actually a really cool example of a spin-off benefit from funding astrophysics research. Maybe you don't care so much about the position of galaxies that you're never going to visit, but you probably do care a little bit more about stopping the spread of viruses, and understanding their transmission helps us do that. Okay, so now we have this really cool technique that allows us to see where the droplets from a cough end up. We can count the number of droplets, we can see how far they travel, we can even see how big the droplets are. For example, in the first version of this experiment, which is where all the pictures I'm showing you today came from, we did 10 coughs in a room with standard ventilation. On average, for each cough, we were able to detect around 25,000 drops of spit over the 10 sheets of paper, which extended over 3 meters away from the cough. And that's a lot of spit that can travel a long way from a cough. The smallest spot we were able to detect in this experiment was about 120 micrometers in diameter, which is just over 1/10 of a millimeter. On the other hand, the largest spot we found was about 24,000 micrometers in diameter. So the range of particle sizes produced in a single cough is incredibly large. There are actually a few simple ways to make the resolution of this experiment better, and in subsequent versions of this experiment, we could actually detect even smaller spots, and I'll talk about this in future videos. However, no matter how good we get our resolution, we're only ever going to be able to detect droplets that fall out of the air in a reasonable time period. We can't detect any of the aerosols produced in a cough, as these form a sort of cloud around you and stay airborne for a really long time. The other thing is that we use the cough simulator, this machine here, instead of a real person. Whilst this provides a great proxy for a real person, people will always want to see experiments like this done on real people. One difficulty about doing that is that a cough originates down in your lungs. So to do this experiment properly, we'd need to get the fluorescein dye down in someone's lung, which is a bit more challenging than just putting it in their mouth. Now it is possible, but it's more difficult, and there are more safety protocols that we need to follow in order to do that. Despite these limitations, this technique has the potential to say some really interesting and important things about the spread of diseases through droplets, including COVID-19. For example, we can and we are using it to study the true difference between being indoors and being outdoors. How much safer is outdoors really when you're talking about droplet transmission? We can also study other things like the effect of different types of ventilation on the transmission of droplets, and also things like the efficacy of different types of face coverings on droplet transmission. Stay tuned on this channel for more on the results of these different ideas being tackled with this cool technique, where astronomy and astrophysics meet epidemiology and medical science. Until then, stay safe everyone. Subscribe if you're interested in seeing more about this, but until next time, I'll see you soon. Bye!